Good evening, everyone. Thanks for coming out tonight. I appreciate this opportunity to speak with you all as part of the Prince William Sound Tuesday night lecture series. So this evening, I'd like to provide a broad overview of avian influenza, or a highly pathogenic avian influenza, or HPAI, which is sometimes also called bird flu. So admittedly, much of what I'm going to speak about tonight is not exactly uplifting information. However, I am hoping that you ultimately walk away from this talk feeling empowered. That is, although HPI is something that we all need to now live with, there are steps that we can all take to be proactive in our response to this emergent wildlife disease. Is going the wrong way.
Try. Sure, it didn't turn on.
There it goes. Thank you. Okay, so here's the outline for this presentation. I'm going to start with very brief introductions of myself, my office, and the laboratory that I lead. Next, I'm going to provide some general background information about HPI. I'll then summarize detections of HPI among Alaska wildlife and some results from recent research within our state. I'll continue by conveying three things that I think we can all do about HPI. And finally, as time allows, I'll take questions from you all. So I'd like to start by introducing myself. So I was born and raised in the Midwestern United States, though I've now spent most of my life here in Alaska. For the past 23 plus years, I've lived and worked on traditional Dena'ina lands in Anchorage, and I've dedicated my entire professional and academic life towards better understanding the health and status of our fish and wildlife resources. So this includes degrees in fish and wildlife biology from Colorado State, as well as veterinary and biomedical science from the University of Georgia. I began my professional career working with U.S. Fish and Wildlife Service in Western Alaska, and have since spent more than 23 years conducting research for the USGS at the Alaska Science Center. Much of that research at the USGS Alaska Science Center has been to better understand avian influenza in wildlife, primarily using genetic and serologic methods. And outside of work, I enjoy hiking, skiing, paddleboarding, wildlife photography, bird hunting, and fly fishing, probably things in common with many of you. So as mentioned, I work for the USGS Alaska Science Center, and the Alaska Science Center is a federal office within the U.S. Department of Interior, where we strive to provide objective scientific data that managers, policymakers, and the public can use to make informed decisions about our natural resources. So I want to stress that the Alaska Science Center is not a management organization, and we do not make any policy or regulatory decisions. So at the Alaska Science Center, I serve as the director for the Molecular Ecology Lab, and the Molecular Ecology Lab provides genetic information on the health and status of our biologic resources. So in the Molecular Ecology Lab, we conduct research projects that pertain to wildlife health and disease, population genetics, and environmental DNA. So we've been conducting research on avian influenza in wild birds inhabiting Alaska within the Molecular Ecology Lab for almost 20 years now. So when we began studying avian influenza viruses in the Molecular Ecology Lab, the ecology of these viruses closely resembled what's depicted on this really busy slide. So I'm not going to cover all this information, but I would like to step through just a few portions of this slide because that is helpful for describing where HPI comes from. So not all influenza viruses affecting birds cause HPI. There are many types of avian influenza viruses that naturally occur among waterfowl, shorebirds, and gulls, and that do not cause disease. Birds usually appear perfectly healthy when they're infected with these types of influenza viruses. And we have regularly found these types of influenza viruses in wild birds sampled throughout our state for many years now. Avian influenza viruses that naturally occur in wild birds sometimes spread to domestic poultry. And usually poultry infected by those viruses appear to be perfectly healthy too. It's only a small proportion of avian influenza viruses that have the potential to make domestic poultry sick. It is a small proportion of viruses that cause severe disease and death among chickens and turkeys that are referred to as HPI viruses. So the tendency for influenza viruses to cause serious disease and death among birds or to become highly pathogenic typically only occurs after viruses from wild birds have been introduced in the poultry. So HPI viruses that evolve in poultry are readily spread among chickens and turkeys. These HPI viruses may result in the death or destruction of really large numbers of domestic birds and are therefore extremely costly to the poultry industry. So for example, since just 2022, more than 200 million domestic birds have been affected within the United States alone. So the spread of HPI from poultry to wild birds, depicted here with the red arrow, has only become common since 2002. That is, historically, HPI was almost exclusively a poultry disease. So prior to 2002, HPI had been detected exactly once among wild birds, specifically in terns in South Africa in 1961. Things have changed. So over the past 2 decades, HPI has become an important wildlife health issue. The HPI viruses recently affecting wildlife are genetically related and descended from a virus that was first described among domestic geese in East Asia in 1996. So although HPI has more commonly affected wild birds for more than 2 decades now, the impacts of HPI on wildlife have been particularly significant within just the past 5 years. That is, since approximately 2021. Contemporary HPI viruses have evolved such they're now readily maintained and dispersed by wild birds, largely independent of poultry. So this is what's facilitated this rapid geographic spread. So this map depicts the spread of HPI over approximately 2-year interval from 2021, to 2023. So at this point, HPI has been detected among wild and domestic animals throughout most of the world, including the Antarctic. The only continent, in fact, on which wild and domestic animals have not recently been affected is Australia. So since late 2021, HPI has spread extensively throughout North America, both via migratory bird movements and also through domestic animal husbandry practices. Within the United States, there have been many mortality events among wild birds and wild mammals since 2021. That is, there have been more than 19,000 confirmed detections of HPI among wild birds in the US, with cases in all 50 states. These confirmed detections among wild birds represent only a small fraction of the true number of birds that have been affected. As per domestic animals, again, more than 200 million domestic birds have been affected by HPI since late 2021, including poultry in all 50 states. In a globally unique situation, the U.S. has confirmed numerous detections of HPI among dairy cattle herds. Detections have now been extensive, with more than 1,000 affected herds confirmed to be affected from 19 different states. And as the number of cases in wild and domestic animals have mounted throughout the U.S., so too have the number of cases reported in humans. The CDC has now confirmed 71 human cases of HPI in the U.S. since 2024, the majority of which have been associated with domestic animal contact. The CDC also reports 2 deaths associated with these cases. So the maintenance and spread of HPI viruses has become increasingly complicated within the US and globally in recent years. That is, there have been many new hosts and routes of transmission recently identified, presumably on account of increased exposures as well as ongoing viral adaptation. Thus far, the situation in Alaska has been somewhat less complicated and in many ways less severe as compared to other regions within the United States and elsewhere in the world. This is at least partially due to the limited production of commercial poultry and dairy cattle within our state. So this slide helps to summarize the current ecology of HPI viruses in wild birds and spillover hosts here in Alaska. HPI viruses are now readily maintained and dispersed by wild birds. HPI viruses sometimes spread from wild birds back to domestic poultry here in Alaska. HPI viruses have also sporadically spilled over into mammals. And HPI viruses have caused some level of mortality among virtually all animal hosts in which they've been detected. So far, mortality among wild birds and mammals affected by HPI has been reported to be relatively sporadic and of fairly low magnitude here in Alaska. So, for example, HPI has been confirmed among a total of 570 birds. More than half of these detections have been among ducks that have appeared perfectly healthy when they were sampled or harvested by hunters. Of the birds in Alaska that have been found to be sick or that have died on account of HPI, bald eagle, common raven, common mirror, and glaucous gull have been the most frequently reported species. Other wild bird species that have repeatedly been reported to be adversely affected by HPI within our state include black-billed magpie, black brant, Canada goose, lesser scaup, northern pintail, parasitic jaeger, Sabines gull, and short-tailed shearwater. HPI has also been confirmed among a relatively small number of wild mammals here in Alaska, including 5 red fox, an Arctic fox, 3 polar bears, a brown bear, a black bear, and an ermine. All of the wild mammals confirmed to be positive for HPI here in Alaska have been reported to be sick or to have died. So for context for those numbers, HPI has caused numerous large-scale mortality events among wild birds and mammals at other global locations. So for example, as part of a mortality event in eastern Canada during 2022, it was estimated that at least 40,000 seabirds died on account of HPI. In South America, it has been reported that more than 600,000 seabirds and more than 50,000 marine mammals have died on account of HPI. So thankfully, we have not yet had observations of such large numbers of mammals negatively impacted by HPI here within our state. There have also not been any human cases of HPI reported in Alaska. So now I'd like to convey some of what we've learned through recent research on avian influenza and wildlife inhabiting Alaska. So I'd like to begin by highlighting an annual sampling effort that we've been conducting in collaboration with U.S. Fish and Wildlife Service staff at Eisenbeck National Wildlife Refuge and with collaborators at the University of Georgia. So since 2011, we've collected approximately 1,000 samples from hunter-harvested waterfowl and wild bird feces deposited in the environment each autumn in and around Eisenbeck National Wildlife Refuge and the adjacent community of Cold Bay. Samples have been tested for all the types of influenza viruses, including those that cause HPI. This annual sampling effort has provided us with a wealth of information regarding the occurrence of avian influenza viruses in wild birds in the environment, and we've used this information to better understand how viruses are maintained and dispersed. So we focus our research efforts on avian influenza in wild birds inhabiting this particular region of southwestern Alaska for several different reasons. First, southwestern Alaska represents an important staging area for migratory waterfowl, where birds come from both Russia and Alaska to congregate during autumn. Second, Eisenbeck Lagoon and the supporting area supports a relatively high level of sport and subsistence harvest of waterfowl. And third, through prior genetic analysis of these influenza viruses, we found that this is a region where viruses may be commonly introduced from East Asia into North America. So now I'll highlight some of our more recent findings from the past several years of sampling. So in 2022, we detected HPI viruses among hunter-harvested birds in southwestern Alaska for the first time. We detected HPI viruses in 54 samples collected during September and October, most of which originated from northern pintail ducks. We also detected HPI viruses from samples collected from mallards, cackling geese, gadwall, American widgeon, green-winged teal, and northern shoveler. Genetic analysis indicated there's at least 5 types or genotypes of HPI viruses in these samples. 2 of these viral genotypes appeared to have been introduced into southwestern Alaska from East Asia. 2 other viral genotypes have appeared to have been introduced from the lower 48 states or from Canada. And the 5th viral genotype we detected had not previously been reported, and it may have emerged within this region through a process called reassortment. And that can occur when a host is infected by 2 different viruses and they swap genetic gene segments. So during autumn of 2023, we detected HPI among only 4 samples collected from hunter-harvested waterfowl within the region. And those four HPI positive samples originated from northern pintails and a single mallard. And genetic characterization of the viruses indicated the recurrence of viral genotypes that we had previously identified. And during autumn of 2024, we detected HPI in 18 samples from hunter-harvested waterfowl, including northern pintails again, as well as green-winged teal. And we also detected HPI in a single mallard that was found dead. Genetic characterization indicated the presence of a viral genotype that we had previously identified in Alaska, as well as another emergent viral genotype. This emergent viral genotype we detected in Alaska during 2024, the term D1.1, subsequently spread across North America through wild bird movements within a matter of months. I've never seen anything quite like this. Viruses of this emergent genotype quickly became the predominant HPI viruses within the United States and Canada, affected countless wild and domestic birds, as well as wild mammals. These viruses were also introduced in dairy cattle and affected a number of humans. So this example speaks to the importance of Alaska as a location where novel HPI viruses emerged and the far-reaching implications to human and animal health. So you might be wondering at this point, what factors influence the prevalence of HPI among wild birds inhabiting southwestern Alaska in any given year, and how those viruses are dispersed across the landscape? So I think one of the most important factors is what's called population immunity. So population immunity refers to the proportion of animals with antibodies to HPI viruses. So these antibodies are produced when animals are exposed to HPI viruses, and they facilitate recovery from infection. So animals with antibodies to HPI viruses have some level of protection against reinfection and may be less likely to develop severe disease or to die if they are ultimately reinfected. So independent lines of evidence suggest widespread exposure of Alaska wildlife to HPI viruses since late 2021, which may be leading to changes in population immunity. So for example, here's a figure conveying the occurrence of HPI viruses of 7 different types, or again, genotypes, that were detected in Alaska during 2022. So this figure was generated by one of my colleagues in the molecular ecology lab, Dr. Christina Ahlstrom. So the height of the bars indicates how many different times HPI virus genotypes were detected, and the colors correspond to the different regions depicted on the map to the right. The timeline on the bottom indicates when viruses were predicted to have been introduced or to emerge within Alaska. So these data provide evidence that HPI viral genotypes that were initially introduced into the state in late 2021 and early 2022 quickly spread, as illustrated by the colorful bars on the left side of this figure. And in contrast, HPI viral genotypes introduced later in 2022 exhibited much more limited spread, as evidenced by the shorter bars on the right side of the figure. So this pattern is consistent with extensive viral spread among a susceptible population, mostly lacking antibodies to HPI viruses, followed by much more limited spread once many individuals develop antibodies and recovered from infection. Additionally, we found evidence that numerous wild mammals sampled in Alaska had antibodies to H5 and N1 influenza following HPI introductions in late 2021 and 2022, which is suggestive of prior exposure to HPI. So animals with H5 and N1 antibodies included numerous red foxes and brown bears, as well as a single Canada lynx and a wolf. These data suggest that HPI infections are not always fatal among wild mammals, and that some individuals actually recover from infection. Furthermore, these data suggest that some species of wild mammals, such as brown bears and red fox, may be exposed to HPI at relatively high rates at specific locations and in specific contexts. That stated, I think it's important to acknowledge that these data may not be representative of all mammals across our state, or even throughout the entire region where they were sampled. It's unclear how mammals with antibodies were exposed to HPI viruses, though the scavenging of sick and dead birds seems like a probable mechanism. So to summarize, recent research efforts on HPI in Alaska wildlife I'll offer the following 5 takeaway messages. First, state and federal government offices within Alaska are actively tracking HPI and conducting research on this important wildlife disease. Second, genetic analyses support HPI virus introductions into Alaska from East Asia, as well as the lower 48 United States and Canada. Third, Alaska's location where novel HPI viruses may emerge with potentially far-reaching implications to human and animal health. Fourth, HPI viruses may be more common or spread more readily among wildlife in some years, and less common or less likely to spread in other years, which may be related in part to changes in population immunity. And finally, HPI viruses appear to be widespread among wildlife in Alaska for multiple years now, and therefore it's unlikely this is going to go away anytime soon. So now I'd like to offer some ideas regarding things we can all do about HPI as it continues to affect wildlife here in Alaska. So I want to start here by being clear and upfront. There is nothing you nor I nor anyone can do to stop the occurrence and spread of HPI among wildlife. However, we can all choose to be informed, to be prepared, and to take action when there are additional detections of HPI in Alaska, or if we witness wildlife that we suspect might be affected. So let's talk for a minute about each of these bullet points. The first thing we can all do is to be informed, right? You're doing that right now. By listening to this presentation, you're going to be able to make more informed choices about your working, harvest, cleaning, and cooking practices based upon your perception of risk. You're also going to be able to make more informed choices about how you're going to respond if you witness wildlife that you believe might be affected by HPI. Identifying wildlife that is potentially affected by HPI is not always straightforward. Some animals, such as ducks, may appear perfectly healthy or asymptomatic when they're affected by HPI viruses. However, many animals affected by HPI become sick and die. This disease has a variety of presentations. The most common signs of HPI among sick wild birds include a lack of coordination, stumbling, inability to stand upright, inability to fly, walking or swimming in circles, a twisted neck, and paralysis. That stated, any animal that appears to be obviously sick, that does not try to escape when approached, or that has died from an unknown cause, should generally be considered suspect for HPI. So there's some great online videos showing behaviors that have been associated with HPI in wildlife. These behaviors don't confirm that an animal is affected, but they're extremely suspect. And the types of behaviors I encourage you to look for and to report if you see them. So let's have a look at a few examples. So forewarning, these videos show animals that are sick and might not be super comfortable to watch. So if you prefer not to see these short videos for the purpose of better understanding what you could encounter in the field, I encourage you to briefly look away from the screen for a few minutes or step out of the room. I'll let you know when the videos are over, and just bear with me for a sec as I get these videos to play. So this first video was made in the Bering Strait region near Golovin in May of 2022, and was shared on the Leo network by Hunter Gray and Gage Sheffield. And it depicts a brant swimming in circles, and swimming in circles has repeatedly been associated with HPI affected birds. So this next video I'm going to play was taken in the Seattle area, obviously of a bald eagle. It was posted to YouTube by Kirsty Mule, who's an urban conservation specialist. And this eagle had poor coordination and was unable to fly. Again, signs that are commonly associated with HPI. This next video is of sick bald eagle that was posted to Facebook by Who's Woods Raptor Center. This eagle was encountered in Iowa and was ultimately confirmed to be affected by HPI. So this eagle displays a twisted neck, often called torticollis, and was readily approached. Again, these are signs that are suggestive of HPI infection. And this last video I'll play was also posted to YouTube. This video shows 6 sea lions in Argentina that were suspected to be affected by HPI. And signs of HPI among affected marine mammals include lethargy, difficulty breathing, and ocular or nasal discharge. Animals may also display a lack of coordination, exhibit tremors, and be readily approached. So to obtain additional information about recent HPI detections in both wild and domestic animals, you can find comprehensive lists of all USDA laboratory confirmed detections online. And to become more informed regarding human health and safety on account of HPI, I encourage you to consult the CDC website. If you are a hunter or if you help to process wildlife that has been harvested, the USDA and US Fish and Wildlife have issued guidance regarding steps that can be taken to reduce the risk of HPI to human and domestic animal health. So suggestions offered by these agencies, including disinfecting your knives, equipment, and work surfaces that were in contact with wildlife during butchering, as well as cooking all meat to an internal temperature of 165 degrees Fahrenheit. And this USDA and US Fish and Wildlife guidance for hunters can also be found online. So once you've become informed about HPI in wildlife, the next thing you can do is to be prepared. So if you work for an agency or a tribal entity, you might check to see if your organization has an HPI response plan. And if so, familiarize yourself with the document. If you're not associated with an organization with a response plan, I encourage you to develop your own personal response plan. So your response plan is going to help guide you if there are additional detections of HPI within the region, or if there's unusual mortality that could be on account of HPI. So to clarify, I'm not proposing that you write a manual. Rather, I'm suggesting that you consider your options for modifying your current practices as indicated by the images on the right. So for example, is there anything about your practices at work or when butchering or preparing harvesting animals that you think might help to lower your risk of being exposed to HPI or the risk of spreading HPI to other areas? Have you carefully considered your practices as far as washing your hands, your knives, work surfaces, your equipment, and your rubber boots following the harvest and processing of animals? Do you have a plan if you observe sick or dead animals? For the latter, I encourage everyone to consider reporting your important observations of sick and dead animals. Your observations may help to raise situational awareness within your community and guide organizational response. So when reporting sick and dead wildlife, it's extremely helpful to include the date and time, specific location, species affected, the number of animals that appear sick and that have died. And taking photos or videos with your phone can be extremely helpful for documenting what you observe. Speaking of photos, please consider taking a photo of this slide and adding these reporting coordinates to your contacts. To report sick or dead wild birds, please call the U.S. Fish and Wildlife Service, Alaska Sick and Dead Bird Hotline, or the USGS National Wildlife Health Center via email. Observations can also be posted to the LEO network. To report sick or dead wild mammals, please contact the Alaska Department of Fish and Game. So once you're satisfied that you've become informed about HPI, after you have prepared your own personal plan for response, you're now ready to take appropriate action. So I encourage you to use information about HPI from credible sources, such as those mentioned today, to make informed decisions about your work, harvest, cleaning, cooking, as well as reporting practices. Also, please remember that reporting observations of sick and dead wildlife may ultimately help others in your community, in your region, and throughout Alaska to make more informed choices. If you participate in the spring subsistence harvest of birds or eggs, there are other important ways you can take action. I'm pleased to convey that at the request of John Whistle, the USGS is partnering with the Native Village of Eyak this year to test birds and eggs harvested this spring for HPI viruses. So if you're interested in having your birds and eggs tested, swab samples will be collected by staff of the Department of Environment and Natural Resources. You can please contact John at the phone number, email address indicated here to get your spring harvested birds and eggs tested. Your birds and eggs will be immediately returned to you following sampling for processing and consumption. The samples collected from spring harvested birds and eggs will be tested for HPI viruses by partners at the USGS National Wildlife Health Center in Madison, Wisconsin. So the National Wildlife Health Center is one of the most established and best recognized federal institutions within the United States for the diagnostic investigation of wildlife diseases. Staff at the National Wildlife Health Center provide services to federal, state, military, and tribal partners to assess the disease status of wildlife managed under federal authority on federally or tribally managed lands. So I've worked with the National Wildlife Health Center for more than 2 decades and can attest to their expertise conducting these diagnostic tests for HPI. I do want to be clear and upfront, though, that samples are not, the testing of samples is not anticipated to be rapid, such that you'll receive results prior to the processing or consumption of birds and eggs. Rather, these testing results will likely be received by staff at the Department of Environment and Natural Resources for the Native Village of Eyak after a period of weeks or months following the conclusion of the subsistence harvest season. So this is on account of the complicated logistics and shipping procedures to maintain a cold chain for ensuring the accuracy of diagnostic tests. So that stated, I hope this partnership will provide your community with contextual information about HPI that you can use for future decision making regarding harvest practices. So sampling efforts in Cordova this spring are going to extend the practice of partnerships between the USGS and tribal entities to test subsistence harvested birds for HPI. So for example, during 2015, when HPI was also maintained among wild birds in North America, albeit at lower prevalence at the time, the USGS partnered with the Yukon-Kuskokwim Health Corporation to test birds from 10 Western Alaska villages during the spring subsistence harvest. Ultimately, HPI viruses were not detected among subsistence harvested birds as part of that sampling effort, which provided information to local residents about their exposure risk for that specific context. So again, if you're interested in having comparable information generated for your community in the context of the 2026 spring subsistence harvest, please participate in this opportunity by reaching out to John at the Department of Environment and Natural Resources and having your birds and eggs tested. At this point, I want to acknowledge the many cooperators that contributed information and data presented this evening. I'm extremely appreciative for those contributions. And thank you all for your time and attention, and I'll be happy to address any questions that you all have. Thank you.
Do you have any data on sea otters?
I have no data on sea otters in Alaska. There was recently a positive detection in a sea otter reported in California.
I know Kate McLaughlin has a question online. I don't want to miss her.
Okay.
Kate, go ahead.
Hey, Katrina Hoffman put a question in before me in the chat, so I don't want to jump in line.
Oh, thanks,
But
Kate.
go ahead, Katrina, ask your question first, then I'll ask mine.
You sort of answered it. There's a lot of trappers in our community, and I jumped the gun just by typing it in earlier. I wasn't sure if I'd be able to stay to the end of the presentation, but are there any additional recommendations you would make for trappers who may be working with live or dead animals, depending on what they encounter while trapping, and who are also typically skinning those animals?
Thank you. That's a really great question. I don't personally have any recommendations I can offer. I would probably start by advising going to the USDA and US Fish and Wildlife recommendations that are posted online. I think lots of practices would be common between hunting and trapping, although maybe not 100% so. So that would probably be a good place to start. And I'm unaware of any specific guidance for trapping. That seems like an important gap. Thanks for bringing that to my attention.
Thank you.
So first off, I'd just like to say hello to a fellow UGA alumni. Good to see you up here north. Graduate in 95, last class of zoology before it went to the Odom School of Ecology. So I'll give you an idea how old I am. I work with hummingbirds, so I'm really concerned about, has there been any, or what is the prevalence of occurrence of avian influenza in passerines? Very few, I would assume.
Thus far, there have been a great question. Thank you for that. Go dogs. So far, there have been proportionally much fewer detections of HPI among passerines. That is consistent with prior research that's shown that other types of influenza tend to be very low among passerines, songbirds. So I believe that is likely valid inference. I have heard voiced numerous times that some people are skeptical. There's a detection probability bias. To translate that, passerines tend to be small birds. You mentioned hummingbirds, they're really small birds. To find a really small bird dead is less likely than finding like a big white swan that's dead. So it's possible that people might not be finding passerines that are sick and that have died as frequently. But there's been a lot of literature, a lot of testing of passerines in the past, and they tend not to be great hosts for influenza viruses. One reason is that they're less associated with water than, say, waterfowl or aquatic birds, and that's the primary mode of transmission, thought to be the primary mode of transmission for most influenza viruses. So I think there's probably something to that. So, your inference is as valid as anybody else's, but there's maybe some thoughts on that.
Great. Thank you for that information. Appreciate it. And thank you for the talk.
Thank you.
Out of the waterfowl, why northern pintail?
I think that is largely a bias just on where we're sampling. So northern pintails are an extremely common duck in Cold Bay, Isabek region, much more so than other species. So I think that's a good part of it. We find a similar level of prevalence, a similar rate of viruses among mallards and green-winged teal as compared to northern pintails too. They're just much less abundant, mallards and green-winged teal as compared to northern pintails. Excellent question.
Next question for John. Are you only targeting the spring outcome? We're starting slow. We're getting something going, trying to sustain it, but I expect we'll be doing this repeatedly. For right now, we're just gonna stand up the spring harvest and then probably talk about the fall. But I think our goal is to have it sort of on its own eventually.
Maybe a question for Brant, were you in contact before? Or is there still a person or a contact if you find something and see something or whatever that you can tell, or do you think it's better just to go to the website that you gave us?
Yeah, that's probably a better question. I mean, I would say talking to Bio and Forest Service or pushing data is a good step, but I would get it directly from Randy and probably give you a message.
Go right to those websites that you gave us.
That's the other.
Oh, go direct.
I mean, most important is just to tell someone and make sure that it gets the people that could help respond, I guess, I would say. So if it's easier for you to tell someone and initiate response, that's the way to go. If it's just easy for you to, pick up the phone or send the email, then that's the way to go. But it's just try to initiate the response, get the word out, help inform the community, and if it's possible, then to, get the carcasses or have someone, follow up with the sick birds to see if you can diagnose that, yeah, that is HPI, or if it's not HPI, maybe it's, harmful algal blooms or something else that's relevant for the community.
And as long as you tell like somebody, we pretty much know, like,
Yeah.
if you call Charlotte, she's gonna get a hold of Caitlin from Marine Mammals or whatever, all the folks that deal with it, so.
Who to call, so,
Yeah, we'll figure it out. Please.
So is there stages, or if I go duck hunting,
For now.
maybe it's not quite,
How many?
you don't always get sick real fast. And then is it okay if I eat it? I mean, I want to go kill something just to let it.
165 degrees.
So thank you, that's a great question. So a few things, like one, everybody's perception of risk is a little bit different, right? And I'm not going to tell anybody, here's what a person should do. And so I think everybody needs to make their own judgment about the data that's out there and then what's a person's perception of risk. I can tell you, we've sampled a lot of ducks that hunters have harvested and that hunters ate and hunters cleaned. There have been no cases of HPI among humans in Alaska. So there's some information there. U.S. Fish and Wildlife suggests cooking meat to 165 degrees Fahrenheit. 165 degrees Fahrenheit will kill the virus. So some more information out there. Exposure and infection is complicated. There's lots of different steps in the process. You have to have exposure. The route and dose of exposure means a lot to development of disease. So I think that is all to say that good handling practices will limit or minimize exposure to. So we talked tonight about, things that you can do in your own plan as far as keeping a clean workspace and wearing gloves if that's where you perceive risk, and bleaching surfaces when you're done. So I think there's lots of different steps that could be taken to minimize exposure as well.
Not good old duck hunt. I was back hunting pheasants last fall and COVID, and there's a lot of snow geese that were different stages, but said they would try to escape from you, sitting there dead. So is it could it be an overpopulation thing too, or?
I think there's a few different layers of the onion with snow geese. So there have been lots of detections of HPI among snow geese throughout the continent. There is a lot of snow geese across the continent right now. There's not been a tremendous amount of good data to quantify the true number of snow geese that have been sick and that have died. So I'm kind of scratching my head about what the real situation is with snow geese. And I wish I had better answers for you. There are certainly a lot of snow geese that have been affected by HPI. There's certainly been a lot of snow geese that have died on account of HPI. The total numbers, I honestly can't tell you.
But when you just see them around, you know, I mean, okay, hey, there's one. Oh, there's another one.
Unfortunately, the way the data is reported, like if you go onto the USDA website, which I encourage everyone to do, just look at, 19,000 detections and to get an idea about the number of taxa affected. So I talked with hunters over the course of the last few falls that would come across fields with many dead birds, tens, hundreds, thousands. I've heard all sorts of estimates. And if 3 are ultimately picked up and sampled and sent into the National Wildlife Health Center, and they do the diagnostic tests on those 3, and they all come back HPI positive, the number reported online is 3 HPI affected snow geese. So that's where I'm struggling a little with how to address a very smart question with snow geese, and that the true number of snow geese affected is much more than going to be on the USDA website for sure. I can't tell you how many more it is, but it's a lot more.
Is there concern about transmission to pets, especially like dogs?
Some people do have concerns about transmission to pets and specifically hunting dogs. There has been some research looking at antibodies among hunting dogs, and there has been limited evidence for exposure of hunting dogs to HPI. I would say there's not a wealth of data out there yet. To my recollection, there have been 2 dogs that I believe there's been 2 dogs that have been confirmed HPI positive, but I think there's been relatively few cases when you think of how many hunting dogs are out there.
Did they survive?
One dog, I believe, did not survive. I believe that dog actually consumed an infected bird. Not sure about the other kids. That's the best of my recollection.
I have a question. Is there a length of time that the meat can be frozen that you no longer have to worry about this?
Freezing, not so much. Unfortunately, influenza viruses can be frozen and be still viable much later. Freezing and thawing, that process of actually freeze and thaw does help to deactivate the virus, but it takes multiple freeze thaw cycles. So I have not seen any recommendations with regarding to freezing and thawing meat. I don't know that you really want to do that. I think the only recommendation I've really seen as far as deactivating the virus is through cooking to 165 Fahrenheit.
And what is the effect on a human, like the people that are infected?
Mm.
Are they surviving this? What are their symptoms?
So the CDC has confirmed 71 cases, I believe, and there have been 2 fatal infections associated with HPI in the United States. Almost all of those cases, almost all cases have been associated with domestic animal contact. And the CDC has a great website. If you just do bird flu CDC, there's pages and pages of information about human exposure to bird flu.
The other animal or bird that we consume, and I guess Santo cranes. Has there been any Santo crane cases?
There have been few cases of HPI in sandhill cranes, but not many confirmed. Yeah, and I do know that from a conservation standpoint, this is something that has been on numerous people's mind, including my own, when you think about like the concentrations of sandhill cranes that occur in the mid-continent at about this time of year, actually, that the number of really large outbreaks we've seen in the US has been pretty modest so far. But then when you look at like what has gone on elsewhere on the planet, there's been some really large outbreaks. And largely when there's really high concentrations of animals. So fortunately, that has not occurred with sandhill cranes. Yeah, maybe just some things to think about there.
Interesting that the passerines aren't very susceptible. What about shorebirds?
That's interesting. I had an email thread today about shorebirds. So shorebirds can be a host of influenza viruses, including HPI viruses. There probably is a detection bias similar to passerines with shorebirds. So there have not been lots of shorebirds confirmed affected by HPI. Maybe different than songbirds because shorebirds have been associated with lots of other influenza viruses. I think from my own personal perception that detection probability bias is probably affecting the number of reports for shorebirds. So I think shorebirds tend not to be as a permissive host of avian influenza as say waterfowl. I think those are tend to be the most permissive hosts, but on the spectrum, they're still a pretty good host of influenza viruses. So there have been few large mortality events in shorebirds. There has been one in the Wattle Sea over in Europe reported a couple years ago, but there haven't been many reports of large mortality among shorebirds. But this is something that I think those working with shorebirds might keep on their radar. And here would be a really good place to keep it on the radar in the next month or two.
Thanks very much.
Please, is this something that's just inherent in birds that's kind of flaring up, or it's something that's?
So this is something I've been tracking for 18 years, and 18 years ago, this was a poultry issue. This was HPI occurred when viruses naturally maintained in species like mallards here would get into domestic poultry, and a small amount of those viruses might heat up and become highly pathogenic, cause disease among poultry. Now those viruses have spilled back over from poultry into wild birds enough times and for long enough that now these 4 contemporary HPI viruses, what we're seeing nowadays, are descended from those poultry viruses and now gone back in the wildlife, and they're really good at being maintained in wildlife now. I don't think they're going away. I think this is their new reality, that now we have viruses that emerged. I saw it over the last 18 years that are here for the foreseeable future that now we have to deal with. That they're fit. They're now they're maintained readily in waterfowl. They're getting into other species of wildlife. I think some of the rates of exposure that we're seeing for other wildlife, wild mammals, are relatively high. I don't see how this disappears off the landscape. I think this is a new normal. It's lousy, but I think it's just what we can do now is be informed, be ready, and take action as appropriate when we see something.
How do the birds catch?
That's a great question, and I didn't do a very good job of explaining that. So influenza viruses and HPI are usually spread via water. So water birds can readily spread it. So as they're swimming around in the water, they're pooping in the water, and that puts the virus out in the water. And then they're also feeding in the water, like ducks. And so then they're picking it up and catching the virus and spreading it amongst them. That was a great question.
Well, thank you.
Yeah, thanks.